Hello everybody. On behalf of 908 Devices, I would like to welcome you to this webinar. The topic of today's webinar is ZipChip, a versatile platform for analysis of small molecules, intact proteins, and beyond. In this presentation, I will first present an overview of the ZipChip technology, explain how the platform works, and then we will go over some key application areas where ZipChip has an advantage over the traditional methods. So let's begin with the overview of the ZipChip technology. What is ZipChip? ZipChip is a capillary electrophoresis based front-end separation device for mass spectrometry. It is a lightweight nanoflow device that automatically syncs and aligns with the mass spec inlet. The ZipChip is currently compatible with several different models of mass specs from Thermo Fisher, Bruker, and SciEx. The second aspect of ZipChip hardware is this microfluidic device shown in the center of the screen, which is also called the ZipChip. It integrates capillary electrophoresis and nano-electrospray ionization on a single device, making coupling with mass spec seamless. There are two types of chips available: high-resolution chips for applications that require good separation for thorough characterization, and high-speed chips for rapid analysis. And finally, we have the ready-to-use reagent kits that cover a wide range of applications. The kits eliminate the need for method optimization and provide a ready-to-use solution for the users. Installation of the ZipChip is pretty straightforward as well. In the image on the left, you can see the user holding the ZipChip interface. It weighs about 8 pounds and can be easily mounted on the mass spec. The image on the right shows the user mounting the interface on the mass spec. The installation takes less than a minute, and after installing the interface, a few electrical and fluidic connections are made. This is followed by priming of the fluidics and a microfluidic chip. And then you can start generating data in less than 30 minutes. Now let's take a look at how the ZipChip works. The schematic of the chip is shown in the center of the screen. The chip has 4 wells, a serpentine channel, and a sprayer corner. The chip performs 3 main functions. The first important function of the chip is sample handling. The autosampler delivers the sample in the sample well. The sample is then pressure injected into this injection cross before turning the power supplies on. Once the high voltage power supplies are turned on, positively charged analytes migrate through the separation channel towards the sprayer corner. This is the second important function of the chip: electrophoretic separations. And the third function of the chip is nano-electrospray ionization. As the analytes make their way to the sprayer corner, due to the voltage at this corner, the analytes are electrosprayed directly into the mass spec. And this is a liquid-only separation, and the channels do not have any stationary phase. This significantly reduces the possibility of injection-to-injection carryover. Because ZipChip uses a microfluidic technology, the analyses are performed in a fraction of the time when compared to traditional methods. In the example shown here, a mixture of 20 amino acids was analyzed on the ZipChip. Most of these amino acids were baseline resolved in less than 2 minutes. Compared to this, when the sample was analyzed on a traditional capillary-based CE system, it took more than 10 times longer for that analysis. The same is true for liquid chromatography separations as well. ZipChip produces results that are much faster than conventional LC methods. So the ZipChip is not only orthogonal to liquid chromatography separations, but it also produces faster results. Now I will provide an overview of the main application areas of the ZipChip. But first, I would like to mention that ZipChip is a truly versatile platform for analysis of a wide range of molecules. The application space ranges from small molecules to peptides to intact proteins and beyond. The platform has been widely adopted in biopharmaceutical industry and leading research labs for a variety of different applications. Just in the last year alone, there have been several peer-reviewed publications highlighting the unique aspects of ZipChip. And some of these publications are highlighted here on this slide. But how does ZipChip simplify the life of an analytical scientist? As I mentioned before, it is a single platform that can be used for a variety of different applications. In most cases, there is little to no method development necessary, and switching between different applications is a hassle-free process. For example, you can run a number of different applications on the same day. You could be analyzing small molecules or amino acids in the morning. You could be running a peptide map on a protein before lunch. And then you could switch to intact protein applications in the afternoon and also analyze native proteins after that. This is because ZipChip eliminates the need for system conditioning before analysis and usually only requires to switch the background electrolyte solution and also the chip type to change application type, offering great flexibility to the users. Now let's take a look at the first application space, that is analysis of small molecules and metabolites. Capillary electrophoresis in general is a great technique for analysis of small polar organic molecules. ZipChip can not only separate analytes based on their net charge, but it can also resolve structural isomers that have the same charge but a different hydrodynamic radius. And this type of analysis is usually performed on a high-speed chip on a ZipChip and requires just a short amount of time. There is no method development required, and a ready-to-use method can be easily adopted. The first example shown here is that of an amino acid separation. This is a promega amino acid standard comprising of a mixture of 20 different amino acids. You can see that most of these amino acids are baseline resolved in less than 2 minutes. And if you notice carefully, the first 3 amino acids have sharp peaks. This is because lysine, arginine, and histidine are dibasic amino acids and have a net positive charge of more than +1 because of an extra basic amino group. As opposed to that, aspartic acid, on the other hand, has a net positive charge of only +0.3 at pH 2.2. And therefore, due to the difference in the overall charge, lysine migrates much faster and aspartic acid migrates much slower through the channel. This is how the charged base separation can separate analytes. But there's also isoleucine and leucine that migrate at about 1.3 minutes. And although they have the same net charge, they're baseline resolved because they're structural isomers and have different hydrodynamic radii in solution. The next example of small molecule application is that of analysis of beta-methyl aminoalanine, or BMAA for short, from shellfish extracts. BMAA has two other structural isomers with very similar properties and pose significant analytical challenge for traditional separation methods. The authors of this study were able to get clean separation of all three isomers in less than a minute, and that too in their very first injection. There was no method development required. And due to this, the overall method time was very quick, and they were able to very quickly analyze a large cohort of samples. There were about 200 samples in just 2 days, when the traditional method would have taken much longer than that. So the ZipChip can provide great separations, but can the ZipChip also provide quantitative information? The answer to that question is yes. Similar to liquid chromatography, the ZipChip can also perform formal quantitation. In this example, several different amino acids in human plasma standard from NIST were quantified, and the data was compared with the values that were provided by NIST. And as you can see from this table, in all cases, the data was not only accurate and fell within the error bars, but the method was also highly linear, over 5 orders of magnitude dynamic range. That shows that the method is very sensitive and accurate. The second application space that I would like to talk about is peptides analysis. The ready-to-use method can be used to determine primary amino acid sequence for proteins at both at the MS level as well as the MS-MS level. In addition, it can also resolve glycopeptides, deamidations, oxidations, and other site-specific modifications from the corresponding native peptides. And as is true with other methods, the analysis times are generally very short compared to traditional methods. A group of analytical scientists at Amgen performed a study where they analyzed trypsin digests of a monoclonal antibody and a bispecific antibody, both on ZipChip MS as well as LC-MS. As you can see from the electrophoregram on the left, the ZipChip performed the analysis very efficiently in less than 10 minutes. The same sample was then analyzed on their nano-LC and took 200 minutes to analyze. The method was not only fast, but also separated critical quality attributes. As seen from the extracted ion electrophoregrams on the right, isoaspartic acid and asparagine deamidation peptides were separated easily from the corresponding native peptides with baseline resolution in under 10 minutes. The method also separated other critical quality attributes such as methionine and tryptophan oxidation very efficiently. The authors also looked at the sequence coverage obtained in both these methods. So the sequence coverage for both the mAb and the bispecific antibody was better in case of ZipChip MS because the ZipChip was able to account for the smaller hydrophilic di and tripeptides, which the LCC8 or C18 column cannot do a good job of retaining. And as a result of that, the authors concluded that the throughput of the MAM or the multi-attribute monitoring method can be increased up to 20 times with the use of the ZipChip, and it can provide results comparable to the traditional methods. In another study, a group of Genentech scientists used the ZipChip for analysis of constrained peptides. Constrained peptides contain multiple disulfide linkages and are often susceptible to forming aggregates due to intermolecular disulfide scrambling and other hydrophobic interactions. And this can complicate the analysis of such molecules. The ZipChip peptides method was easily able to separate the peptide aggregates from the peptide API in just 8 minutes. The method also revealed that two different peaks were observed for the dimer, which suggested that the sample likely contained structural isomers of the aggregates also. Similar separation was not observed on the LC, which highlighted the advantage of the ZipChip in analysis of constrained peptides. The ZipChip can be easily used for analysis of native proteins. The chip is capable of transporting native proteins while folded and also electrosprays them, enabling generation of native mass spectra. The method is simple, does not require any cleanup, and is compatible with formulations containing detergents. The users can easily and quickly observe modifications such as deamidation, C-terminal lysine clipping, glycosylation, etc. at the native protein level. An added advantage of this method is that it produces separations that have similar peak profiles to other complementary separation techniques such as capillary zone electrophoresis with optical detection, CIEF, or cation exchange chromatography. The electrophoregram shows the analysis of the NIST map using the native charge variant analysis method on the ZipChip. The method uses native antibodies background electrolyte solution and a high resolution native chip. The sample was directly diluted from the formulation buffer and loaded on the chip. The separation produced several different features. There was a main species, two basic species, and a cluster of acidic species. The mass spectra for each of these species is shown on the right. The mass spectra confirmed that the protein is still folded. The zoomed in view of the spectra focusing on a particular charge state is also shown, and it shows that the different glycoforms that are present in the sample. The method is highly sensitive, and with just 1 nanoliter injection volume, it can positively identify species that are as low as 0.1% relative to the main species. The exact same method can be used for analysis of a wide range of antibodies. As shown in the electropherograms here, several different antibodies such as Herceptin, USP-MAB, Cytopoint, and cetuximab were analyzed using the same separation and MS method that was used for the NIST map. The only difference between these methods is that they used different analysis times. The proteins migrated at different times depending on their isoelectric points. Thus, this method can eliminate the long method optimization times required by conventional methods, freeing up the precious instrument time. The most recent addition to our applications toolkit is the oligonucleotides analysis application. Oligos are single or double-stranded polyionic molecules that have properties that are much different from the traditional small molecule or protein-based drugs, and therefore they pose a different set of analytical challenges. The most widely used method for characterization of synthetic oligonucleotides is reverse phase HPLC coupled with mass spectrometry, but using ion pairing agents. The ion pairing agents such as hexafluoroisopropanol or alkylamines can foul the mass spectrometers and also the LC systems, and it can take a long time to decontaminate them. One of the major advantages of the ZipChip method is that it eliminates the use of ion pairing reagents, making switching between methods fairly easy and fast. Just like other applications, the method does not require any method development and can perform analysis in short times. The ZipChip oligos method uses a chip with a different chemistry. While the other ZipChip methods that I showed use chips with polymer coating on the channels, The oligos method uses a bare glass chip with no coating. This allows for the negatively charged glass surface to repel the negatively charged oligonucleotides, and therefore the analytes migrate down the separation channel due to high electroosmotic flow in the bare glass chips. They produce sharp and Gaussian looking peaks. Separation of 5 different 20-mer single-stranded DNA molecules is shown in the electrophoregram on the left. All 5 peaks were observed as positive ions and were detected with excellent mass accuracies. The positive mode separation generates much fewer charge states compared to negative mode, improving the sensitivity of the method. I would like to point your attention to 2 specific peaks in the electrophoregram, peak 1 and peak 4. The only difference between these peaks is that peak 4 has a DNA with an additional phosphate group that adds an extra negative charge, which causes it to migrate much slower. And this highlights the charge-based separation aspect of ZipChip. The oligos methods is very sensitive and can detect a 20-mer down to about 10 nanomolar concentration. The method is highly linear over 4 orders of magnitude dynamic range and allows for easy quantitation of oligonucleotides. The robustness of this method is shown on the right. The same sample was injected multiple times and produced peak areas and migration times with low RSDs. The migration time RSD was found to be 1.6% and the peak area RSD was 13%. And for comparison, these numbers are very similar to other ZipChip applications as well. So in conclusion, the ZipChip has several advantages over traditional separation techniques. The platform seamlessly integrates capillary zone electrophoresis with mass spectrometry. The microfluidic technology used in the ZipChip can provide huge improvements in MS workflows. It can eliminate sample preparation in a lot of cases, and also method development. It can improve the throughput by 20 times or better. It consumes only a tiny amount of sample, and especially for the oligos method, it does not require the use of any ion pairing agents. The method is very versatile and can be used for analysis of several different biomolecules, including small molecules, peptides, proteins, as well as oligonucleotides. Thank you for your attention.